Hello, welcome back to another, not Battle World, another science stream. Hello, George. Yes, we are here today to discuss immunoglobulins. As for the jumping jacks set 4, does my mic work? I think it works, right? Yeah. As for the jumping jacks, I will do that after about an hour because I literally just ate and I do not want to reverse the flow of food. Okay? Okay, the mic works. Let's go. So, one moment. So, regarding... Where is my marker? This is the marker. Immunoglobulins. Globulins. This one, we're talking about the genetic diversification of it. Alright? And these are the Y thingies, antibodies that you see, and it has different types. So just as an overview, I think I'll talk about this in a bit, but I'll try to summarize this in a very quick sense. These are proteins used to help you defend against pathogens or bad stuff. So, and they're made up of 2 amino acid chains. This is the heavy chain, and these small ones are the light chain. And those Y things, if you just make them like go like this or this way, the antigen from the bacteria would be around here or something, or the virus. Essentially, the bad stuff would be there, and then this side would be your like the B cell, or essentially this is you. Okay. That's how it functions in a very, very nutshell way. Where's my eraser? Okay, there it is. And for its types, you have this one. You have this is the IG A T E G M with the is also the IG. This one, these 3 would be single Ys. This one, the A is usually a dimer. Oh, I'll just draw it properly. Bam, bam. And dimer means 2 of them, like together, like that. And then M, it's single if it's at the surface, but normally it's a pentamer, 5 Ys. A lot of Ys. 1, 2, nope. 1, 2, 3, 4, 5. This is the biggest one. The most common one is IgG. The least well-known, the not famous or not as studied enough is IgD. So that's how, that's what their differences are. Okay. Alright. So next, okay, now let's do the very, very specific stuff about it. Erase. Okay, so we have the variable and constant regions. That's the one that changes depending on the antigen. IgD, the D, wait, it was A D E G M, right? D, it wasn't studied enough, but it's still functional. Meanwhile, A is more common for like the respiratory stuff. I'll talk more about it. That was just a very big overview for it. But yeah, that's the general antibodies. For now, let's go over the adaptive immunity. So the lymphocytes, those rearrange their receptors to participate in different immune responses. And then you have the B cells and the T cells. Okay, another familiar term. I did not watch the Cells at Work anime. I only watched a couple of episodes, but I did not finish it. But I think that's a good show. For B cells, these neutralize pathogens and stimulate the macrophages. I really feel like these were mentioned in that anime. I should watch it. Meanwhile, T cells would kill the infected cells. So again, B cells neutralize pathogens. Meanwhile, you have T cells, two types, I think. You have helper and cytotoxic. Cytotoxic would be the killers. Helper would be the ones that release different stuff to stimulate more things. Yeah, but that anime should be watched by numerous people. And this, okay. Yeah, you have B cell, T cell, cytotoxic T, regulatory T. These are the lymphocytes. I will draw more of it here, because this will help. This mini figure will help. You have B lymphocyte, you have helper T lymphocyte, and then you have cytotoxic T lymphocyte, and then you have the regulatory T lymphocyte. Alright, B lymphocyte would neutralize the microbe. Helper T lymphocyte, helper T cells, activates the macrophages because they're helpers. They activates macro and do inflammation as well. And activate the B cells. Cytotoxic, who are they? They're the killers. Kills infected cells. Cytotoxic, toxic. Regulatory would suppress the immune response. That's why they're regulatory. So again, B cells, and we have 3 types of T cells right now. So B cells would neutralize the microbe. Helper, cytotoxic, and suppression. Helper would activate the B cells, and it would activate the macrophages and inflammation. And then for cytotoxic, they kill the infected cells. Regulatory would suppress the immune system. Alright, good. Then you have the primary and secondary response. Erase. Then now you have, again, this one. Primary and secondary response. So, when you get exposed to an antigen, the immune system would do, hello Elmore, welcome back to the stream. We are talking about, not amino acids, we're talking about antibodies. So for antibodies, if you get exposed to an antigen, the immune system would mount a primary response that is quite slow. So this one is slow. And it would produce the antibodies with moderate affinity. So just kinda effective. Not as much though. But at least it's there. And then, if the antigen is encountered again, the bad thing, if it sees it again, that's the secondary response. And it's now very rapid and, oh, I wrote it at the wrong. Oh, black holes. That's a good question, but I did not prepare for that question right now because we are talking about molecular and cell biology. So, I'm afraid you're at the wrong stream. But for black holes, I think they're essentially, okay, they're powerful things. That's one thing. The second is there's a part there where it's a point of no return, where if you go, I don't remember how much distance you have, like how far you have to be there, but there's a part where if you go in, you can't go out anymore. So So there's that. So you have to be careful with that. But there's a thing where if you're already at the point of no return, once you realize you're at the point of no return, you're screwed. You can't go anymore. You can't leave. So yeah, you just have to accept your fate. But I think the nearest black hole is not really that near, hopefully. Yes. What is that? Yeah, maybe. But hopefully, it would still be more than a gazillion years before Earth gets affected by one. So yeah. So that's one thing. That's one less thing to worry about. One thing we have to worry about is the secondary response, because, well, we don't have to worry if it's secondary response because it's rapid, and now those antibodies would have a stronger affinity. So they work much, much better. So again, primary is when you're just okay. Let's say you get a cold or the flu, like that. No, the flu might be a bad example. Let's just say disease A. Yeah, the primary and secondary response. Oh yes, that is true. How many times? Yeah, it's like, imagine if it's I was gonna say Gojo. Which one? Eren. No. It's Kakashi trying to discipline the students, wherein okay, Kakashi and Naruto. Kakashi would be the immune response, and then Naruto's like, Kakashi sensei, I want to eat ramen again for dinner. So the primary response of Kakashi is like, no, it's kind of slow for now. Secondary response would be bring out the chinelas and then bam, like that. So yeah, that's the primary and secondary response. Secondary response is much, much, much quicker and much more effective. The structure, I already gave a small sample of it, but I will mention it here again. So you have the Y. This one. The Y. Beautiful drawing. This one. Wow. Oh no. That looks so bad. One moment. I'm gonna draw it again. This one. I'm gonna be confident with the angles. Oh no. You know what? One more time. I'm gonna write this part. And here. Alright. Okay, beautiful. That's the heavy chain. And then you have the small light chains over here. Nice. Nice. So, oh, I should just draw it in a different color. Never mind, I'll erase it again. I'm gonna use blue for the heavy chains. Blue. One more time. Drawing stream. Okay, this one. Nice. One shot. Heavy chain and then light chain. Nice. Wow, that looks so beautiful. Alright, you have the antigen binding site here. Antigen binding site. Binding site. And then these are the light chains. Light chain. And then you have the heavy chain. Heavy chain. And then you have the transmembrane region. It would just be there. That's your membrane. The transmembrane region. Transmembrane region. Okay. And these are variable regions, aka they change. The pixel perfect lines. I know. Look at that. That's pixel perfect. The variable regions would change depending on who they want to target, but your constant regions are constant. Oh, by the way, for the antigen binding site, I was gonna say you have the... Oh, no. This is your antibody, right? Next, where where will the antibody like go? So in the bacteria, there in your antigen. No, there's like the T cell receptor. I think at the bottom. No, I won't draw it yet. I'm not sure about where this one is. But yeah, this is your antibody. T cell receptor could be later, but it has alpha chain and your beta chain. So the highest variability in antigen receptors would lie in their complementary determining regions, aka the CDR3, and the regions are formed within the very stable immunoglobulin fold. Okay? And then again, if you consider the lock and key concept, it's similar here. It's very similar here. Let's see. There you go. No. Alright. Normal color. Normal color. You have this. Let's say you need a circle there. Then you would put a circle here. But then if this is a triangle, then you need a triangle here. So that's kind of how the antigen antibody binding is also. The pocket can accommodate various antigen shapes. So yeah. And lo and behold, SARS-CoV-2, aka the virus that causes COVID-19, because SARS-CoV-2 is the virus. SARS-CoV-2 is the virus. Meanwhile, COVID-19 is the disease. That's the difference. Do not mix that up if there's a multiple choice question. Alright. So antibodies can neutralize viruses like SARS-CoV-2 by binding the specific epitopes on the viral spikes protein's receptor binding domain. Essentially, it can neutralize SARS-CoV-2. That's why. And you have, I talked about this earlier, the different binding types, the IG, A, D, E, G, M. IgA forms dimers, DEG, single, M, single or pentamer, 1 or 5. B cells would first, produce IgM during the initial immune response. Kinda slow, but and then using like some mutations, like somatic hypermutations, the variable regions would switch around. Yes, that's how it works. So again, the B cells would go under VDJ recombination, okay, to create the primary response, primary number 1 after and then the stages. You have the stages, different stages of the B cell. Let's redraw this one. The B cell development progresses from stem cells. Okay, that's a lot. Let's see. You start from the stem cell stage. These are the stage of maturation. You start from the stem cell, and then you become the pro-lymphocyte. Oh, they look alike. I won't even draw it anymore. Pro-lymphocyte. And then pre-lymphocyte. And then immature lymphocyte. And then mature lymphocyte. And then differentiated. Essentially, stem cell to lymphocyte to differentiated effector lymphocyte. Now, there is also the part about your immune system and the lymphoid tissues. Right. Let's do that one. The immune system, you have the primary and secondary lymphoid. So immune system. you have primary and secondary lymphoid tissues. Primary would be your bone marrow and thymus. And those are the sites where lymphocytes develop. Lymphocytes develop here. Where is my sticky note? I need to sticky note this part. Found it. Dun, dun, dun. Sticky note. So, primary lymphoid tissues, those are your bone marrows and thymus. Lymphocytes develop there. And then you have the secondary lymphoid tissue, aka the lymph nodes. Lymph nodes, your spleen, and etc., where the mature lymphocytes would encounter the antigens. So sometimes when you go to the doctor, they would like tap the thing. That's what they're tapping, trying to check if it's inflamed. So they're checking if you have some kind of inflammation and if there's something going on with you. If I were to draw, I will draw it. Haha. I will draw a human. 1 second. This is your human. That's the nose. Wow. Okay. And then this, that's the arm. Nope. It was alright. This is not a titan. Okay, that's the human. This is the arm. Look at that. Okay, nice. Very nice. So that's the human. You have the bone marrow. Let's draw the bone marrow here. You have the bone marrow here. Yup, beautiful. That's the bone marrow. The bone marrow would deliver stuff to the lymph node. Oh no, not yet. This is your heart. The heart. Where's the spleen? Not yet. Then you have the lymph node. I'll just draw them all in the same way. Yeah, this is Picasso level. Van Gogh. That's the heart. And I'll draw green for lymph node. Green, not red. Green. That's the lymph node. Lymph node. And then you have the spleen over here. Nice. So the lymphocytes go from the bone marrow, travel there, and it gets this delivered here. You have the Peyer's patches somewhere there. And then we'll also go to the spleen. Also goes to the lymph node. Essentially, the bone marrow and, again, bone marrow and thymus, those are your primary lymphoid tissues, while your spleen and lymph nodes would be your secondary. That's where your mature lymphocytes would encounter the antigens and go bam, bam, bam. That's the oil rig, where it goes bam, bam, bam. Now, we're gonna talk about... Yes. We're gonna talk about... Oh, really? It's scared. They're trying... Really? I wonder why. It's scared to trigger the COVID PTSD. Yeah. Okay, now that's... Next would be the lymphoid. I drew the heart earlier because the lymphocyte circulation and normal circulatory system is very related. Lymphocyte circulation and immune surveillance. Immune surveillance. Surveillance. Valence. I cannot. Surveillance. No. I will. One moment. Why is this word is weird. Immune survey survey surveillance valence. Nice. Okay, so the reason why they're intertwined is because Lymphocytes continuously circulate between the blood and lymphoid tissues back and forth, so that if there are antigens floating, they can find it. They will... Wait. Water. Really? And it was trying to automatically block the message. Yeah, they're avoiding the COVID PTSD. But we have the, what's this called? Primary and secondary responses for it. And get vaccinated so that it won't happen again. Lymphocyte circulation, where was I? Oh! Okay, let's draw another human first. This is a human. Why am I drawing it that way? It's alright. Picasso level. Yes. Nice. Very nice. Very nice. Look at that. That's a human. So, oh, I was gonna draw the subclavian vein. So I need it to be bigger. Human-chan. Human-chan. Hi. Nanigaski. Here. Okay, I'll just write this part. Nice. Okay. And now the legs. Short legs. Okay, beautiful. So now you have the heart here, and then at some point over here you have the subclavian vein. Subclavian vein. Left. The left subclavian vein. So, you have arterial blood, right? Blood that came from the heart. Then you have venous blood. Blood that's going back to the heart. And then you have the lymphatics. Because they're going... The lymphatic stuff will go back inside through the subclavian vein. Bum bum. Right? Yeah, enter lymph nodes. No. 1 second. Lymphocytes circulate between blood and lymphoid tissues. Naive lymphocytes enter the lymph nodes via arterial blood. Yes. And survey for pathogens presented by antigen presenting cells. After antigen recognition activation, lymphocytes exit through the efferent lymphatics and return to the bloodstream via the thoracic duct and travel through sites of infection. Okay, we have the naive lymphocytes, the baby lymphocytes, enter the lymph nodes via arterial blood. Okay. Essentially going back and forth. Left subclavian vein. Pathogens from the site of infection reach the lymph nodes via lymphatics. I know. It's like, which way are they going? It's okay. Just don't mind the subclavian vein. Just remember that the lymphocytes and the lymph return to the blood via the lymphatics. There's the canal. And the pathogens from the site of infection reach the lymph nodes via the lymphatics. That's how it works. They talk to each other. Okay, now, oh, this one's messier to draw. Because this is the cellular traffic in a lymph node. Wow. But it's gonna be fun. It's alright. Let's draw it. Let's draw it here. Oh, there you go. Here is your dendritic cell. Oh my gosh. Okay. This will be whacked up. Let's just write the lymph node first. Efferent vessel. Okay. That's a bit tame. So within a lymph node, this is the lymph node. It's trying to drain an infected tissue, aka like lymph node trying to drain to drain an infected tissue. Alright? And then you have the dendritic cell carrying your microbial antigens. Okay? You enter via the afferent lymphatics. Alright? So the dendritic cells come in. Dendritic cells. I know. But this is what's happening inside the body. Dendritic cells carry the microbial antigens, enter through the afferent lymphatics, and interact with the T cells. Now they talk. The dendritic cells talk to the T cells, T cell area, where the activated helper T cells would engage the B cells and lead to the differentiation. Okay. Essentially, macrophages would, this is the important one, macrophages eat the bacteria. While, oh yeah, this one, macrophages eat the bacteria. Meanwhile, plasma cells from the B cells would exit via the efferent. So plasma cells are going out. They're your extra guards. Plasma cells exit through here to release the antibody, to secrete antibodies. Because they're like, oh, we see the bacteria. We must go and and bring more soldiers outside to defend the body in case there's more bacteria coming in. So essentially, there are attackers here, and then they're sending more troops out. So that's how they work. Macrophages eat the bacteria, and then the plasma cells would, what's this called? Would go out through the efferent vessel and secrete antibodies. You see, they're useful. They're useful. Then you have the germinal centers. What are they? They are structures that arise from a few activated B cells. What? Okay, germinal centers arise from a few activated B cells. You have the dark zones, and then yeah, I have to draw this one. Whoops. There you go. You have the dark zones here. Erase. Alright. So I erased that one. Germinal centers. Centers are dynamic structures that arise from B cells. From some B cells. You have the dark zone. Let's just write it this way. Dark zone, where B cells called centroblasts, or times 2 times 2 times 2, they accumulate there. And the light zone would be where those cells become like centrocytes, interact with helper T cells. So from centroblasts to centrocytes, and then those would then interact with T cells. So dark zone to light zone, centroblast to centrocyte, and talk to T cells. Following activation, the IgM positive B cells migrate into the germinal centers. There, it's like a house. It's like a house. Okay. Oh, for immunological memory to be effective, the antigen receptors must be clonally expressed. This means that each B cell or T cell expresses a single receptor specificity. So it just depends on the lock and key again. Now, where is it? You have allelic exclusion ensures receptor homogeneity. What? Only one allele of the immunoglobulin heavy and light chain genes would be expressed. This ensures that B cell receptors on a given cell are identical in antigen specificity. If allelic exclusion fails, B cells may express mixed receptors. Oh, okay. Essentially, it just wants specificity, aka the 1:1, and not necessarily 1:2. just so it has a clear job description. Yeah, clear job description. And now we have the VDJ recombination, variable joining, and diversity segments. Like VDJ, variable. What's the D? Wait, joining, diversity segments. Diversity segments. It's just they have a lot of variability within them because there's a ton of things they need to target. Then you have the RAG 1/2 they form a multi-protein assembly in the DNA junctions. Yeah. And there's the hairpin complex. I think that's fine. And you have an enzyme called Artemis. That's a good enzyme. Okay, this enzyme asymmetrically opens the hairpin from earlier, which would create some overhang, and you can add a couple more stuff there so that it's even more diverse in form. Because after the overhangs are created, your DNA strands are aligned and joined. So it's good to know. Good to know. By the way, VDJ it's in a very tightly regulated timeline, because if that's screwed up, then your B cells and the other stuff is also screwed, and you just don't want to die because of that. Alright? Now, in the bone marrow, oh, there's a good one. In the bone marrow. In the bone marrow, immature B cells that don't bind self-antigen would mature and begin expressing both IgD and M. So B cells would be IgD and IgM and would exit to the bloodstream. But if they bind to a self-antigen, it is retained inside the bone marrow. Okay? RAG expression is upregulated, and then the cell can attempt to rearrange it. That's what happens if it's retained in the bone marrow. BM, bone marrow. Nice. Light chain. The evolution, we have evolution of the immune system, the adaptive immune system. It is centered around the emergence of VDJ recombination, and that's because of the RAG1 and 2 genes. And these genes are only in jawed vertebrates like mammals, birds, reptiles, and fish. Now, you have a slide. No, we have a slide called somatic hypermutation. Okay? So mutation would be, I think this one's a good lesson. Mutation. It is any change in the sequence of genomic DNA. Alright? So these changes can include point mutations like insertions, deletions, duplications, or terminal events like gene conversion. And in immunoglobulin genes, your somatic hypermutation would refer around to the non-templated changes. So just remember mutation, any change in the sequence of your genomic DNA. It can be an insertion, deletion, duplication. With insertion, you just add something. Deletion, you remove something. Duplication, you times 2 something. Point mutation, single nucleotide substitution. Dun dun. Note. Somatic hypermutation would occur at very, very defined hotspots in your immunoglobulin genes. And that's how they are very specific. You have lesions as well. Like the uracil can be recognized and excised by UNG, the uracil DNA glycosylase. And you have the cell cycle phase also. I think, oh, I haven't yet. I will be discussing the cell cycle at some point. But the cell cycle phase influences repair outcome because when uracil lesions are processed in the G1, that could lead to gene conversion. Okay? And if because if those bad stuff, if those lesions would persist into the S phase, the error prone thing would continue, and that's bad. We don't want that. So that's why you have to fix it in the G1. Are these mutations something that can just randomly happen to people, or just during conception, or both? I'm inclined to say that it's very random at both, I think. But that's also why some people would try to do genetic sequencing, like if the compatibility of some people would match more, because there are some certain blood types which may, like, as blood, not just the AB and the ABO, the AB combination, there's also the RH factor, the plus and the minuses. So that those different combinations, I forgot which ones are the bad combinations, but that's why you have to get like checked to make sure that those combinations are all right. So because that's also why there are couples who are like, oh, be careful with your first child or something like that, and that's why they have to go to the doctor and get that certain injection or something, or a medicine, to make sure that the child is all right. Yes, that one. So and there are also some mutations, for example, induced, like like the sun would, and this is why you have to wear sunscreen and stuff like that, because if you put just forgetting the exact pathway, but putting some of your cells get stressed, and that's some of those mutations at some point lead to the skin cancer and all that. So that's why, summary, wear sunscreen, or summary, go get genetic counseling. That's the thing. So yeah, it happens randomly to people, or because of some certain situations or certain pairings. Yep. Good question. Keep that up. This will help, because I need to yap for a thing, and I need to practice yapping. Now, those lesions, I talked about the different outcomes and the cell cycle. All right. Oh, and take note, the cell cycle is also happening always, because wait, I was gonna say it's always happening because you make like cell 1 would become like the next generation of cells, and it's really, really quick. So at some point between those times 2, there might be one clone there that's kind of wrong, and usually the wrong clones would get degraded. But if they're not found, then they're not found, and they just keep swimming there. But at some point they are found, so that's why you have to keep your body healthy. Then you have your base excision repair, the BER, which would recognize the uracil DNA glycosylase, other stuff. And then you have the mismatch repair. So these are, so see, you have the different DNA repairs that would help your body stay healthy. Next. Oh, I talked about the hotspots. That's good. That's good. Modeling. Oh, where is this? Okay. UG lesions created by AID can be resolved through replication-driven transitions in phase 1 of the somatic hypermutation. I missed out where. What was the AID? I don't know if this is the AIDS. No, that's not it. That's a different one. Actually, where are the questions? Correlates. Okay, I have reached the questions part. How would you experimentally demonstrate that VDJ requires the 1223 rule? And what would you predict if you mutate one of the RSS? What is the 1223 rule? What? Google time. One moment. Side quest. What is the 1223 rule? So, oh, okay. The 1223 rule. I have to draw this one. Draw it, draw it. Go time. You have the 1223 rule, aka recombination. No, it's not that one, sadly. This one is about recombination can only occur between gene segments flanked by signal sequences with 12 and 23 base pair spacers. Okay. Again, this is about recombination. The mixing up can only occur between gene segments flanked by recombination signal sequences with 23 and 12 base pair spacers. Wow. And how would we test it? To test it, we can use an in vitro recombination assay where different pairs of VDJ segments would be flanked by like 12 or 23 or other spacers. And we could check how efficient that those are, the exchanges are using PCR or a Western blot. Because if you mutate one of those, the recombination should be lessened because it's not the 1223 anymore. And how could you determine whether AID activity leads to a bias towards transition to the GC base pairs during SHM? Whoa. We could isolate the base pairs from wild type and AID deficient mice. Okay, you get the B cells from. Okay, I'm gonna draw this. This will help us see the big picture. This will help us see the big picture. So you have the wild type and where is it? AID deficient mice. Get their B cells. Get the B cells after immunization, and then you sequence the V region and compare VDJ mutation patterns, and you would see that in the wild type, the C and G's would most likely become T and A. Okay? But in the AID deficient one, you don't get the switcheroo. Again, that's how you determine if the AID activity is important towards transitions at the GC base pairs. If you see the switching, if you don't see the switching in the thing without it, then it must be important. Wow. Amazing. Now, how could you detect antigen-driven selection during SHM in germinal center B cells? Okay, we talked about how in the germinal centers, wow, the heart rate is at 83. That's nice. It's not the 80, it's 80. Oh, that's nice. I'm calm, very calm. Awesome. Breathe. Now, let's go. Where was it? How can we detect antigen-driven selection during SHM germinal centers? We sequence the VDJ regions from B cells sorted from the light zone. Light zone, that's the central something. And then the dark zone is centroblasts. Oh, centrocytes, centroblasts, those things. And then you calculate the silent rotation mutations. Dun -dun. Now how? Okay, design an experiment to assess whether receptor editing occurs in autoreactive immature B cells. we can use a transgenic mouse model to express a known self-antigen and self-reactive thing. Again, we isolate things from the bone marrow. One moment. Time to draw. Drawing time. Isolate from the bone marrow. Here we go. Okay. We get, where's it? Isolate, we have the bone marrow. Bone marrow. We isolate the B cells. Immature B cells. Immature, the baby B cells. Mature B cells. From the bone marrow and access the expression of RAG1 or RAG2 via RT-PCR. So if you see, okay, here's the thing. If the receptor editing is active, we should observe the RAG expression. Because if the editing is active, then you should see it. But if not, then it's not there. It's a... Ugh. Now, how could we determine... Oh, that's fine. Ah, what experiment could demonstrate... 78, look at that! Okay, what experiment could demonstrate the role of chromatin looping in immunoglobulin locus contraction using or during VDJ? We could perform FISH. Or chromosome conformation capture in the pro B cells versus non-lymphoid. Okay? And in B cells, just observe the spatial proximity. And... okay, we did that. So for the... wow. For the genetic ones, we finished that. But before I move to the next mini lesson, which is the Golgi, the protein targeting, I will do the jumping jacks. Because it has been about an hour since the food has been digested. It's been digested, right? So far, okay, just to as a throwback, we recently hit 12,000 subscribers. Whoop whoop! And I was like, you know what? To celebrate, why not do 1,200 jumping jacks? Jumping jacks. And I was like, it's gonna be crazy if I attempt to do 1,200 in one day. So I was like, maybe just split them up. Just do 120 during Hello Decipher. Welcome back to the stream. We just finished one lesson and I'm about to do a second, that one to the fourth set. So wait, I'm just explaining what's happening right now. I was like, do 120 in maybe each day. And so far we've done sets instead of day one. Let's go with set. We've done set 1, 2, and 3. How many is that? 360. And we're gonna do set 4 right now. Before I move to doing protein targeting. Crazy. Okay, let me just 1 second. Alright, let's go. See, look at the heart rate. It's calm. It's very calm. Not anymore. Soon. Okay, 1, 2. Wait, stretch. Boom. Stretch. Fighting. Alright, let's go. And 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23, 24, 25, 26, 27, 28, 29, 30. So that's 30. Keep counting. 1 second. So that's 30. 30, 30, 30. I'll just write 30. We did 30 so far, so that I don't forget. Okay. Next. 31, 32, 33, 34, 35, 36, 37, 38, 39, 40, 41, 42, 43, 44, 45, 46, 47, 48, 49, 50, 50. That's a lot. Okay. 1 second. I think the max heart rate we have is like 163, and my goal is just to not slowly but surely we have to lessen that, because that's the way to check if we're becoming healthier, right? Because that means our heart is becoming more efficient. A resting heart rate of just some athletes have very low resting heart rate. So that's the goal. Okay, we're at 50. Let's go. 51, 52, 53, 54, 55, 56, 57, 58, 59, 60, 61, 62, 63, 64, 65, 66, 67, 68, 69, 70. That's 70. Alright. 70. 70. I was gonna get the a boom arm mic thing so that I can move it closer. For example, during like treadmill stuff. But delivery was like, not yet. Anyway, next. 71, 72, 73, 74, 75, 76, 77, 78, 79, 80, 81, 82, 83, 84, 85. Good question. I think these have become a tad bit, just a tiny bit easier. Because for example, the max is 140 right now. At least it didn't hit 163 compared to before. Plus, okay. Plus I learned to take some breaks between them. Yeah. And not to just brute force it. And use the stairs when you can. Alright. 86, 87, 88, 89, 90, 91, 92, 93, 94, 95, 96, 97, 98, 99, 100, 101, 102, 103, 104, 105. Wow. 105. You know what? That's a lot. A little bit more. Okay. 105. And let's go. 106, 107, 108, 109, 110, 111, 112, 113, 114, 115, 116, 117, 118, 119, 120. Whoop whoop. We finished that. Alright, I'm just getting the chair. One moment. See? We did it! We did it! Heck yeah! 120. Whoops, no. 120. Dun 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 dun. The light stick emote is so kawaii. Alright. So that's the update. Oh my gosh, where is my writing? 100. We did set 4 today. So it's now 480. Yeah. Beautiful. And now we drink water. Tubig. Dali. Alright. Next lesson. Protein targeting. That means I will just change the thumbnail tomorrow. I was planning on doing protein targeting tomorrow. I was like, you know what? Why not now? Let's go. Let me just erase this one. One moment. Protein targeting. This one, this might be a lot. But it's okay. We have to get through this. Strong. Be strong. 108. Alright. Protein targeting time. Protein targeting. And let's start. So, in eukaryotes, the whiplash from the jumping into science. Yeah, that's what we do. Imagine if I just pull up Palworld right now. That would be crazy. Crazy, right? Nah, not yet, not yet. We have to finish this. So yeah, we have poor protein getting to be targeted. Of course, it's the new target right now that we're using. So in eukaryotes, most proteins found in organelles are made on cytosolic ribosomes. So, okay, examples would be, okay, the new update. I know it's gonna be tomorrow. Tomorrow? Time zones are weird. It in about close to a list about 23, less than 20 hours from now. The waiting room is up there. It's gonna be there. Yeah. Whoop whoop. And I'm planning on exploring the new islands after I do protein targeting and Golgi apparatus, because there's a thick list that I have to finish. So, in eukaryotes, most proteins found in organelles. Yes, the Cypher, there's a new Palworld update. Have you seen it? I think, yeah, was it yesterday? Yeah, there's a new update where they have a collaboration with the Terraria devs. Terraria. It's spelled Terraria. I didn't play it before. That's why I can't say it properly. But that's the new update that they have. And I haven't found all the islands. I kept on seeing the water. And I cannot fish. Apparently we can fish, but I've never fished in Palworld. Yeah. So it'll be fun. It'll be fun. Join us on that stream. It'll be fun. I will try to fish. I will try to fish. All of the fish ran away last time. AKA 24 hours ago. So organelles containing proteins, aka would be the ER, the mitochondria. Wait. The mitochondria. The nucleus. The lysosomes, the peroxisomes, and the chloroplasts. It's a lot. Oh yes, the new update isn't exactly for like just the older players. They also made sure that the new players could play it because they have the low, like small level pals. I was expecting it to be like Sakurajima and Faybreak, where it's like high tier islands. But surprisingly, it's very accessible. Very accessible. Especially, you know, if I try to go from zero again in Palworld, it might make things easier. Yeah. I totally get it. That's why we're switching around with, you know, the science and the fun stuff. And we're making the science fun too. Because it's a part of life. That's why we're an Isekai'd Mask Knight Empress Scientist. Have I introduced myself? I forgot to do the introductions. Hi, I'm Millennium, Isekai'd Mask Knight Empress Scientist, and today we are going to discuss protein targeting and translocation. Yeah, dab. I cannot dab, the arms won't move. It's just the hair. Even if I do this or that, it's just the hair flip. It's alright. So the mechanisms of protein transport into certain organelles, they are really being studied, including in the ER and the mitochondria and the nucleus. And Those organelles serve as model systems for examining the protein transport strategies used by the cells. Okay? And the mechanisms of protein import into mitochondria and the chloroplasts are similar. But I don't think humans have those. Draw PNG arms. That would be funny. If we draw arms, this one or this. Nope. What are those legs, bro? Beautiful. Both mitochondria and chloroplasts contain their own genomes and ribosomes that synthesize proteins. And protein export from bacteria would resemble protein import into the endoplasmic reticulum in many, many ways. Alright? In many ways. Oh, yep, here's the drawing. I'm gonna draw something now because it's gonna be transport between organelles by the vesicles. Here you go. Okay, so let's say that the whole thing is the cell. I'll just draw the cell. This is the cell. And nope, never mind. We need more space. Drawing a circle is hard. Here you go. And nice. And then you have the nucleus. What's inside the nucleus? Yes, this is the nucleus. What's here? The DNA. Bam bam. Okay. So where's step 1? Let's see. You have the ribosomes over here. They look like burgers. So I'm gonna draw burgers. Because you have that subunit. And then you have the mRNA here. And then you have the proteins being made like that. Yup, beautiful. I think it would be easier if I just draw them in different colors. So I'll just draw the ribosomes in black. Burgers. And then red. The mRNA would be red. And then the growing protein is blue. Nice. Okay. So, step 1, the cytosolic ribosomes. It's called cytosolic ribosome because it's a ribosome that's not stuck in the ER. It's a cytosolic. It's in the cytosol. The liquidy stuff here in the cell. Yeah, cytosol. So first is, I actually saw the DNA is wrapped around these little orb things. Then those orbs are scrunched up into the chromosome. Very, very good. Because the ones you saw, those are histones. Those are histones. Would be kind of look like burgers too, but they're not the same burgers. They would be 4 stuff. And then they would wrap around these like that. And then the little circles would wrap around each other like the telephone coil. So that's how the chromosome is formed. Good. Spreading the science. Very nice. Yeah, that's how you form it from... That's how you form the DNA from just the normal... It goes from the normal line that you know. Like people would say the TTAGC and stuff. And then the 2 layers now, like AATCC. Because it's like A to T, G to C. And then they would coil into these, the coil thingy. That's how the ladder looks like. And then coil them a little bit more, but around the histones that George mentioned, you go like that. And then you coil those things together within each other until you form the X, and the X is the chromosome. So that's how you go from DNA to chromosome. Take note if people say like the words like genes, those would just be specific sections of the DNA, like this one. That could be a gene. This could be a gene. The gene 1 and gene 2. And then if people say alleles, they're just, let's say this is allele number 1 in that gene. In a different like squiggle, that could be allele number 2. So it's just a different type of like thing for that. Alright, so that's how you go from DNA to chromosome. And there's also the word genome, which is just the whole thing, all of the chromosomes as well. Dun dun dun. And we erase it. So now, yes. And now we are there. I erased it. Beautiful. So now we're talking about cytosolic ribosomes. Because we go from DNA to mRNA to proteins, right? And then mRNA is translated by ribosomes in the cytosol. Some proteins remain in the cytosol while others either are directed to the organelles. Okay? So I'm gonna draw one. I'm gonna draw one of these. And since it'll be tough trying to switch around each time. Nope, I'll just switch around each time. I was gonna say, because the colors will help our burger, mRNA, and protein. You have the ER sequence over here. That means they need to go to the rough ER for further processing, and that's why they enter the secretory pathway. So this is your ER now. That's the ER, and your ribosome would go like that, and then your mRNA is, whoa, color please. mRNA would go this way, and then your protein with the ER sequence, nope, blue would go like this, and then you have your ER. There. Now, once they're inside, they would let go of that orange thing, and at some point, they are modified, and then they go to the Golgi apparatus, just as the proteins. This is the Golgi. They would travel, and then the last part is they can go and float to the plasma membrane, or the lysosome to get degraded. Plasma membrane for secretion, lysosome for degradation. And then you also have, I mentioned earlier, yeah, they really help distinguish the names to the drawings because sometimes they look alike. The squiggle A looks like squiggle B, or burger A looks like burger B. No, these proteins I mentioned, the ones just for the cytosol. There, this one and this one. Then you have the E. Nope, no ER because it's not for the ER. Those would just be released as a cytosolic protein. Floats around. And then you could have a targeting sequence. If it has, for example, if it has a different targeting sequence, let's make it purple. Then it might go there. So the nucleus. Water. Wait lang. Water time. Alright. They could go to the nucleus. Or they can go to the mitochondrion. Mitochondrion, familiar? It's a singular mitochondria. Mitochondria is plural. Mitochondrion, singular. Uno, isa. Ichi. One. Okay? Or it can go to the chloroplast. Alright. So those are the mini steps. Proteins are synthesized It's systematic. Very good question. It's systematic. For example, I'm gonna change it into yellow. So you have like the ER sequence. That means, oh, I have to go to the ER. And then you also have specific sequences which may tell it to go to the nucleus. So that's why it helps. I think, yeah, in the next parts we will be talking about the how to distribute them. I think so. Yes. Yeah. Proteins synthesized in the cytosol would be targeted to specific organelles depending on the signal sequences. Nice. And okay, I have a summary like in that I would like to get, but I'll go through this one first. This one is very very detailed, but I'll summarize it after. I'll get that document in a bit. If unless I need to rest the throat, and then I'll continue later. I've really realized that I think it was it Koyori or Bibu who can yap for 10 to 12 hours. Their vocal cords are built different. I noticed that for example there's a difference between like the science streams versus Pal World. I noticed that for the science streams, the way it's like there's more talking, right, in the science streams. Meanwhile, in Pal World, there's a lot of pauses, for example, because of the background music or the pals talking to each other and something like that. So that's why I noticed that. That's why I drink a lot more water, especially in the science streams, because I noticed that it's drying. Like the vocal cords dry out faster for some reason. I don't know. Is it because I'm just it's more continuous? Is that why teachers wear those mic things? So that they don't need to like talk loudly sometimes. But yeah. But don't worry. Wow. 8 hours after the what's this? After that unarchived, was it April? The unarchived karaoke stream? Bro, I could not talk after the I think 1.5 hours. I was like, nah, I didn't really expend my voice for like 3 days after that. So that's why. But the next one I've researched, and I think I will be able to include music next time at least. I'm just gathering some a list of anime songs possibly, and other stuff. That's why. Plus schedule too. I'm trying to see which schedule will work. But yeah. So for the general concepts of protein targeting into organelles, we have to there is Okali also talked about the possibility of not a new group exactly, but because she said, oh, they still don't know if they're really cooking, but yeah, there's a video I saw about it. It's like, oh, there, that would be nice. I think it was Justice, the latest team group that they released for the EN, at least I think so. Okay, so we have the signal or sorting signal. You have the machinery, and you have the energy requirement, the conformation during the transport, and the mode of transport. So those are 5 stuff we have to remember depending on how they're delivered. So for example, proteins can be directed using target or signal sequences, including the amino acid, like this one, amino acid, carbohydrate, or phosphate. And then they often have the Kdel sequence, the lysine, aspartate, glutamine, leucine. Oh no, I have to find the document about the 20. Yeah, Kdel sequence. Again, lysine, glutamate, lysine, aspartate, glutamate, and leucine. Draw time. Lysine. What does it look like? Lysine structure. Bring back the 20 amino acid stream. I have to get that. Oh, lysine is... Alright, let's see. Lysine looks like this one. The COO minus, H3N plus, H, and then you have CCCC and H2. That's lysine. Lysine and aspartate, glutamate, leucine. Okay, but that's the signal sequences. Machinery, the transport, those would be the specific transport proteins that would help. These are the cars. And those are located in the cytosol, organelle, and lumens. Energy could be ATP, GTP. And for the conformation, it could be folded or unfolded. The mode could be co-translational or post-translational. Okay? Okay. And now we have the thing called secretory pathway. We talked about this earlier. It's responsible for directing proteins to their appropriate destinations, including secreted proteins like insulin or luminal proteins of the ER to the Golgi, to the integral membrane proteins and lysosomal proteins. And in the final secretion, they fuse with the plasma membrane either continuously, like constitutive excretion or exocytosis, or in a very regulated manner, like exocytosis again, or regulated secretion, like in hormones. So the flow, we have the flow here. Flow. 1 second. Here. Okay. So you have... we could go from the bottom to the top. Let's do that one. It will look weird, but that's how it looks like here. So you have the rough ER. Squiggles. Squiggle, squiggle. You have the rough ER. You have the squiggle proteins. And squiggles. The burgers are here. The burgers. Burger, 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 burger. Burger, burger. These are the proteins. After the rough ER, they could go through the cis-Golgi network. Cis-Golgi. ER to Golgi to form the cis-Golgi. Note that you also have the retrograde form where the proteins go from this one or this one. But normally just look at this part, the ones going up. The focuses go up. Yeah, this is cis-Golgi network. And then now you have the medial Golgi. And then you have the trans-Golgi. Okay? And then after the trans-Golgi, you have the trans-Golgi network. You now go through the secretory vesicles. Secretory vesicles. And after that, you have the plasma membrane. Plasma membrane. But note, up here, you can actually split, okay? You can go do the regulated secretion or constitutive, because regulated is for plasma, but constitutive could be where is it? That's for collagen and serum. So it depends on the type of proteins that you have and where it wants to go. Oh, and there's a third branch here. Could go to the lysosomes section. And earlier, I talked about this one. I'd briefly describe the burger, the structure of the burger. Over here. Burger, burger, burger. Because, alright. So, you have the eukaryotic ribosome. Okay? That's the ATS ribosome, which is composed of the 40S subunit and the 60S subunit. I know, it doesn't make sense. 40 and 60 becoming 80? Don't ask me why. That's just how it is. And what's worse is the 40S subunit contains the 18S rRNA and about 33 proteins. The math is not mathing, but it is what it is. And then the 60S is, guess what? It's composed of, it gets worse, don't worry. This is a 5S, 28S, 5.8, and about 50 proteins. These don't add up to the thing. But just remember, 40S, this is the 40S small subunit, burger small, and then 60S large subunit. That's the structure of a ribosome. It's weird, but that's how it looks like. Okay? And then we have, what's this called? We also have, let me just draw this really quickly. We also have the protein synthesis and its relation to targeting and secretion. You have polysomes or polyribosome. These are multiple ribosomes translating the same mRNA simultaneously, and they appear as beads on a chain. They're just burgers connected to each other. Many burgers, okay? And for the ribosome locations, you have free ribosomes, which synthesize the cytosolic, nuclear, and peroxisomal proteins, and mitochondrial also. And then you have the membrane-bound ribosome in the rough ER. Because I told you earlier, ribosomes could be free agents, and sometimes they're also stuck in the ER, and that's where they do their thing. So yeah, and just remember, what's the function of a ribosome? They make proteins. Because if there's something you want to remember, DNA transcribed into RNA, mRNA, and then it's transcribed into protein. Who does the mRNA to protein? That's the ribosome. Dun dun. Okay. But yes, I think that is all for today for now. Yeah. Let's do the dab thingy. But yep, thank you for watching. Bye.